The endosymbiotic theory is sometimes called the symbiogenesis theory, and it describes how eukaryotes may have evolved in a prokaryotic world. This idea of evolution was championed by Lynn Margulis in her 1967 paper on the origin of mitosing cells in the Journal of Theoretical Biology. Her paper suggested that mitochondria in plastids like chloroplasts were the evolutionary leftovers of an ancient symbiosis between ancestral eukaryotes and prokaryotes. She proposed that eukaryotic flagella arose from symbiosis between a eukaryote and a spirochete. Now, Lynn wasn't the first to have this idea. She was just the first to present a unified theory that everyone could support. In this video, I'll describe the endosymbiotic theory and provide some reasons for its support. Your objective is to be able to describe the endosymbiotic theory of eukaryogenesis and provide 3 potential sources of evidence. This is a potential essay question on exam number 1, so you want to be sure that you can explain this theory and provide at least 3 pieces of evidence before you take the exam. I want you to close your eyes and imagine an ancient Earth where we basically had a hot chemical soup. Somewhere in that hot chemical soup there were little pools of water where molecules came together and eventually became things like RNAs, proteins, DNAs, and lipids. And one day, these chemicals came together in just the right combination that the first single-celled organisms could arise. We think this happened some 3.9 billion years ago, and in this early Earth, there's no oxygen. So that means that all of these organisms are very simple and they are anaerobic, able to live and make energy in the absence of oxygen. Over evolutionary time, we think that photosynthetic organisms evolved. So these were probably cyanobacteria, which are also prokaryotes, and they would have been anaerobic. However, these types of microorganisms are capable of photosynthesis. They take carbon dioxide, add it with a little bit of water and energy to make glucose. And the byproduct is oxygen, which they would have released into the soup that they were swimming in. Eventually, the amount of oxygen in that soup would have dissolved with it would have dissolved with iron, producing rust. And when the levels were high enough, some of that oxygen would have escaped the soup and gone into the atmosphere. This would have been fatal for many of the anaerobic prokaryotes that would have been on the face of the earth. The only ones that would have survived to today would be the ones that could somehow sequester themselves away from the oxygen. We think that oxygen levels increased in the atmosphere from about 2.7 to 2.2 billion years ago. And oxygen, believe it or not, is a dangerous compound. Many organisms would not have had the ability to neutralize toxic oxygen. So again, many organisms would have died. The oldest fossils of eukaryotes are about 2.1 billion years old. So how did these organisms develop from prokaryotes? Again, the idea is symbiosis was responsible. This is a process in which a unicellular organism, this would have been an ancestral eukaryote, would have engulfed another cell, an ancestral prokaryote, which would have lived inside of the eukaryote. We think that over evolutionary time, remember we're talking a couple of billion years, the endosymbiont, that prokaryote living inside of the eukaryote, lost a lot of its bells and whistles to live independently, and it developed to become an organelle. So without a Wayback Machine, we don't really know that this is true, but this is what we think happened. This is the idea that Lynn proposed. So you start out with an ancient ancestral prokaryote, and one day its plasma membrane starts to fold in. Now we know that cells do this, right? This is basically what phagocytosis is. So the plasma membrane starts folding in, and little bits of plasma membrane probably sealed together to form more complex membranes. You might have seen this before. Remember, the plasma membrane is kind of like olive oil. If you have 2 drops of olive oil on a cutting board and you move the cutting board, those 2 drops will come together and become one large drop. So it's not out of the realm of possibility that these plasma membrane pieces that have folded into the cell would have sealed together and made complex membranes. We think that this is the origin of your nuclear envelope. So when these membranes coalesced around the DNA in the middle of the prokaryote, that would have made a nucleus. So now we have a eukaryote. The different folds would have also led to the development of membrane-bound organelles like your endoplasmic reticulum, your Golgi apparatus, your lysosomes and peroxisomes, things like that. Now, what Lynn proposed and others have proposed is that somewhere in evolutionary time, this ancestral eukaryote now engulfed a prokaryote, but it didn't digest it. Instead, this prokaryote came to live inside of the eukaryote. Now, we think the first one was probably an aerobic bacterium, so it would have been some sort of prokaryote that has figured out how to use the oxygen that's now in the atmosphere to make ATP and drive its energy needs. So these 2 cells, the prokaryote and the eukaryote, live together. The prokaryote likes this arrangement because it's now in a stable environment in the cytoplasm of the eukaryote. And in that stable environment, it has access to nutrients, a stable pH, water, whatever it needs to survive. The eukaryote likes this arrangement because now it has something to do with that oxygen that's in the environment around it. You know, prior to this, there was no chemical reaction really for that oxygen, but now with the prokaryote in there, we can do something useful with the oxygen. We can make ATP. So they live together. Similarly, a photosynthetic cyanobacterium was engulfed by this ancient eukaryote, and these 2 live together. Again, the prokaryote likes being in a stable environment with access to nutrients, okay, and protection. The eukaryote likes this environment because, well, now we've got an organelle inside of the cell that can make glucose, and the other prokaryote, which is now a mitochondrion, can use that glucose after some chemical reactions to make ATP. So everybody starts living happily together, and the result is an ancestral photosynthetic eukaryote that is the forebearer of plants and other organisms. So this theory is the culmination of thought from a number of very smart scientists, but without a Wayback Machine, we don't know if it's true or not. That said, if we compare the characteristics of prokaryotes, eukaryotes, mitochondria, and chloroplasts, it starts looking like there may be some truth to this theory. Here's a table that summarizes some examples. Prokaryotes, as you know, are typically about 0.2 to 2 microns in width, and they can be anywhere from 1 to 10 microns in length. Chloroplasts and mitochondria are 1 to 2 microns in size, width, and length. So that fits within the size for a prokaryote, and it's a size that's distinct from that of your eukaryotes, which are much larger. Your mitochondria and chloroplasts have their own DNA, and it's a circular DNA. What else do you know of that has a circular DNA? Your prokaryotes, right? eukaryotes have linear DNAs, but prokaryotes have circular DNAs, so there's a similarity there. Mitochondria and chloroplasts have their own ribosomes as well. And these ribosomes are 70S style ribosomes, which is more like your prokaryotic ribosome than your eukaryotic ribosome. So because mitochondria and chloroplasts have their own ribosomes, they can make some of their own proteins. An organism that started out independent would be able to make its own proteins, right? Prokaryotes make all of their own proteins. So that's another similarity. Mitochondria and chloroplasts are able to reproduce independently of the cell that they are in. So you might remember, for example, in A&P, you probably learned that when you exercise a lot, the mitochondria in your muscle cells begin to do something like binary fission to become more numerous to support the ATP demands on your muscle cells. Why do they do that? Well, if they had been a prokaryotic organism way back in evolutionary time, most of your prokaryotes do binary fission. That's how the cells reproduce. So it wouldn't be unusual or unexpected to see that mitochondria and chloroplasts do binary fission like the prokaryotes do. Eukaryotes do mitosis and meiosis, which is a very different process. Finally, the DNA in the mitochondrion and chloroplast is not in any kind of nuclear membrane. It's just floating in the cytoplasm, kind of like what we see with the DNA in prokaryotes. So those are some similarities. One other similarity is mitochondria and chloroplasts are double membraned structures. So let's think, if this is our prokaryote, this is the eukaryote that's phagocytizing the prokaryote. Once that prokaryote comes into the eukaryote, we would expect it to have a little bit of membrane from the eukaryote outside of its own membrane, which is prokaryotic. So we would also expect the composition of these two membranes to be different because they're coming from very different organisms. If you look at the structure of a mitochondrion and of a chloroplast, both of those organelles have an outer membrane. And I'm only going to draw the mitochondrion because I am pressed for time to get this lecture done and not drag it on too long. In any case, mitochondria have two membranes. You have the outer membrane, and then you have a highly folded inner membrane where a lot of the ATP synthesis happens. You probably remember this from your Gen Bio. So we have two membranes, which you might expect, and these two have different compositions. Perhaps indicating that somewhere in evolutionary time, our mitochondria were derived from prokaryotic cells that were engulfed by eukaryotic cells. So in this video, I've described the endosymbiotic theory. Again, the idea is that some ancestral eukaryotic organism engulfed prokaryotic organisms and they lived together because it benefited both organisms. Over evolutionary time, the engulfed prokaryotes, one of which would have been an aerobic, capable of aerobic cellular respiration, the other one would have been potentially photosynthetic. These prokaryotic organisms lost a lot of the bells and whistles that allowed them to live independently, so they are now part of eukaryotic cells. I've also provided a table that shows similarities between mitochondria, chloroplasts, and prokaryotes and distinguishes them from the eukaryotes. This provides a little bit more evidence that makes it seem like the chloroplasts and the mitochondria are indeed derived from prokaryotic organisms. You'll need this information for exam number 1. You might try practicing writing your essay, describing the theory, and providing three lines of evidence. If you have any questions, please feel free to reach out to me. Otherwise, I'll see you in the next chapter where we'll start talking about microbial growth.